Hello there, everyone. So tonight we have the great pleasure and honor of hearing from Emily Jones. So Emily is a professor at the Center for Brain and Cognitive Development at Birkbeck. Her research interests center on understanding the cognitive and neural mechanisms that drive variability in developmental trajectories. In this context, she runs a number of prospective longitudinal studies of typical and atypical neurodevelopment in infants and directs electrophysiological and eye tracking acquisition across several large-scale European and global health studies of children and adults with neurodevelopmental conditions. I will hand over to you, Emily. Would you like to share your slides?
I will do. Thanks, Astrid.
Great.
And let me know if at any point you guys can't hear me or if anything goes funny.
Great, thank you.
Brilliant. So let me just share. Oh, that's the wrong button. Share. Okay. Can you see that okay?
Yeah, looks good.
Brilliant. Great. So thanks for that lovely introduction. I'm very excited to be talking to you all today. So what I'm going to talk about is how in our research program we've moved away from thinking about these sort of more deficit-based narratives around neurodevelopmental conditions like autism, which dominated our field and other fields for a long time, and how we're moving towards more neurodiversity-affirmative ways of understanding individual children's developmental trajectories. So I'm going to talk a little bit about the work that we've been doing and some of the empirical data and sort of theoretical work we've done to try to move in that direction. And as I was saying to Astrid, one of the things about moving in this direction is that the stories that we tell with our data get a lot more complicated and a lot less simple. So as you'll see as I go along, I think, reflecting some of the complexity that's out there in children and the way they develop necessarily means that it can be a little bit more difficult to express some of the patterns that we're seeing in a simple way. So yeah, we'll see how it goes. I'll do my best. But just before I start, to say thank you to these people and many others who've been involved in collecting and helping us analyze the data. And you'll see some of them as I go through my slides. We have incredible students and colleagues who work with us, and of course the families who've participated in our studies and our funders. So I'll start by just talking a little bit about neurodevelopmental conditions, why we study them, and then how the neurodiversity kind of perspective intersects with some of our theoretical frameworks and ways we think about development, and how that's led us to come to this new way of studying the early development of children with neurodevelopmental conditions. I don't know, wait, well, hopefully that won't keep doing that. Then I'll talk a bit about how we're starting to think about the long-term dynamics and the short-term dynamics of development and how that fits into those frameworks. And then briefly some implications or, things that might be relevant to policy and practice. So we know that neurodevelopmental conditions like autism and ADHD impact anywhere between 4-10% of the world population, depending on what prevalence estimates you look at. Of course, they're much better detected in some places in the world than others, but we know that they exist worldwide. And we also know that neurodevelopmental conditions come with a range of co-occurring conditions or traits. So ADHD is common in autism, but anxiety is common, depression, other things like OCD, sleep problems, which will come up later. So when we're thinking about the development of children with neurodevelopmental conditions, we want to think much more broadly than just the traits that define those conditions, to think about the range of different outcomes that might be relevant for those children and are often particularly relevant to their quality of life and how they're going to do later on. And we know, if we think about autism, but also ADHD, that the etiology is strongly genetic, so both conditions run in families, but the genes that underpin both conditions tend to be very complicated. So there are some, large effect size changes that will often affect a child's cognitive development and motor development, and also, autistic and ADHD related traits. But for most children with neurodevelopmental conditions, they're probably coming from the combination of lots of genes with small effect sizes that impact a whole range of different pathways, often in brain development, but that include these different areas here. This is from 2018, so this has been substantially expanded upon since then, but broadly a story is similar, that, you know, you get these lots of different neurobiological functions impacted by genetic changes that all have a very small effect in and of themselves. And so there's a very complicated picture, basically, in terms of the genetic etiology of both conditions. And the sort of traditional approach to trying to understand how that might map onto what we see in a child, the difficulties they might have, is to take children or adults with a diagnosis, to look at their brain structure, brain function, and to attempt to link that then to what might be going on synaptically, what might be going on genetically, and to sort of assume that all these things are concurrently related to each other. But we know, of course, in reality, that those genetic changes are present at conception and then are influencing brain development from the very beginning. And there's actually this series of cascading interactions whereby those genetic changes impact synaptic functioning, so the proteins in the synapse and how they communicate with each other, and that impacts how the brain grows and develops. And as the child begins to process the environment, which of course starts prenatally even, that's going to impact the way they process information. It impacts their behavior, so how they seek to take in information, how they interact with other people. And in turn, those things are going to then influence their brain development, which in turn can influence synaptic connections and functioning and have downstream effects on genetics through epigenetic modification. So you get this series of cascading interactions over developmental time that ultimately result in these behavioral changes or mental health changes that we associate with neurodevelopmental conditions. And so to understand what's contributing to that and how we can support children's development, regardless of their neurotype, we want to understand something about this cascade over time. And broadly, what we try to focus on is understanding the pathways that might be particularly important. Because I said, you know, there's a whole range of different genes that are going to be impacted. Together, they're going to probably create this kind of developmental topography. But what we're interested in doing is finding markers of these sort of large pathways whereby once children start to develop down a particular road, you get these sort of self-reinforcing processes, theoretically, that mean they end up in one developmental outcome or another. And if we can understand what marks out those paths, we might be able to figure out how to support children who've gone in one direction or another direction. And so the research design that I'll mainly talk about today, that we've been running for a while now with others, are these studies of infants who have a family history of a neurodevelopmental condition. And so we know, because autism is very heritable, and similarly, I think that the figures are similar for ADHD. If you have an older sibling with autism, you have about a 20% chance of developing autism yourself. In the longer term follow-up studies we've been doing, the rates have become even higher. So 30 to 40% of children by mid-childhood have a diagnosis in our sibling group. So we know that there is a greater likelihood that infants will go on to have at least significant autistic traits. And so we follow groups of infants with a family history of autism and a comparison group who don't, from early in infancy or sometimes prenatally through the infant period to the age at which we can make a determination as to whether they meet a research criteria for autism. So a lot of the studies I'll talk about, we followed them to age 3, but as I said, now we followed them a little bit further on. So one interesting question will be to what extent our results start to change as we start to look at those children who have that later diagnosis. And so in that infancy and toddler period, we take lots of different measurements, genetics, epigenetics, but then measures of behavior, so how the child interacts with people, what their cognitive skills are. We use eye tracking, which I'm sure lots of you guys are familiar with, where we measure what they're interested in looking at on a screen. And then measures of brain development, so EEG and NIRS, which measures blood flow in the brain, essentially, so spatial information about which areas are active, and temporal information about how fast different areas respond. And measures of physiology, heart rate, so, you know, how much a child is excited or worried or anxious about things they see, these physiologically. And then measures of their environment, so their parent their parent-child interaction, but also some measures of the physical environment, but I won't talk much about that. And then what we're trying to start to do is put all this information together to try to understand how these different levels sort of interact with each other over developmental time in the emergence of autism or ADHD. And so one of the things that has become, so over the last sort of, I guess, 10 to 15 years, or 20 now, I guess, that we've been doing research in this way, this idea of neurodiversity has become more and more compelling. And so this is the idea that, I mean, I suppose the truism that there is a spectrum of variation in human neurocognitive functioning. And so rather than think about neurodevelopmental conditions as, in inverted commas, diseases or disorders, we should be thinking about them as differences, and as part of this broader spectrum of ways in which people can differ from each other. And one way some people like to think about it is that autism and other neurodevelopmental conditions reflect just differences in the way people's brains are wired. Another sort of more developmental way to think about it, I guess, is that, yeah, you've just got a difference in where you're going down that topography. So there's a combination of factors that mean you develop differently. But they're, broadly speaking, you know, these kinds of diversity, neurodiversity, like other aspects of diversity, enrich society and bring collective advantages to the group, but also should be valued independent of that. And there's lots of different models in which one can think about this. So the sort of social relational model is one that, you know, we can recognize that impairment and difficulties and challenges can be barriers to thriving. And so that's where we're thinking about supports and interventions and things that we can do. But that also stigmatizing attitudes and social reactions can also cause disability in and of themselves. And so one thing that we need to ensure that we as researchers and as practitioners do is make sure that the research we're doing isn't contributing to that stigma and those, you know, negative ways of viewing people that in and of themselves are going to contribute to disabilities. So more recently, we've been trying to ensure that what we're doing in this field is really pushing in that direction. So that the paper just below is something that we did in collaboration with some ethicists led by Arianna Manzini, thinking about how we can make sure that this sort of translational developmental neuroscience work is done in an ethical and an affirmative way. And so what I'll try and talk about today is kind of I guess the way in which we think about the data and how some of it can be interpreted in that format. So one of the things that we're interested in with this sort of cascading framework is this idea that children increasingly direct their own learning. Of course, it's very familiar to us at CBCD and you guys too. This idea of neuroconstructivism, that children are active learners and seek out what they need to grow and develop. And we know that, you know, you can set out your beautiful learning activity, but depending on what the child wants to do, it's not necessarily going to go that well. And so when we're thinking about some of the differences we see in these longitudinal studies with infants with later autism, we need to be thinking about what changes we're seeing are serving an adaptive function for a child. So when we see differences in behavior, where are they helping the child cope with other difficulties they might have or other differences they might have? Or where might they be maladaptive? So something that the child's having to do to respond to an environment they're in, but that we could mitigate or support them with by changing that environment. And so in these longitudinal studies, we need to start to figure out how we can dissect those two things. And of course, one thing that's really important in trying to think about this is when we're thinking about what might predispose to one outcome or another, in knowing that one way in which we can think about which of those outcomes is positive or negative for the child is through input from people with lived experience. So either autistic people, parents of autistic children, etc. And so there's a lot of effort to do that. So this is from a survey from Autistica, who are a big autism charity, about what the priorities for autistic people are. And, you know, they're in a number of different areas. Communication language is important, but also anxiety. And that's something I'm going to come back to in a minute. So, you know, thinking about the ways in which people define positive outcomes is really important when we're then trying to work out what is adaptive and what is maladaptive for a particular child. So the data that I'll talk about today comes mainly from our STAR study that we run here at Birkbeck. It's also called Basis for anyone who's familiar with it. So as I said, infants with a family history with autism versus those who don't. And we have a number of different visits over developmental time. And then when children get to 2, 3, 7, we one thing we can do is look at the children who have autism versus those who don't, and then look back at the data we collected earlier to determine what precedes that. But we can also look at a range of different dimensional outcomes around things like well-being or their adaptive skills, their everyday behaviors, to try to understand a broader range of outcomes. And I said that really. Okay, so when we started this this kind of research and as a field in general, there was a big focus on social behavior, and this came from, you know, broadly speaking, that sort of deficit-based framing, the idea that, you know, autism is associated with deficits in social functioning, and so what we should be looking at is social differences early on, and maybe these differences will then, in a sort of simple fashion, lead to later social problems. And so we and others looked at, you know, to what extent do we see these differences in social behavior? When do they start? This is some work that Georgia Boissou led, looking at measures of, this is a measure called the Vineland, which asks about everyday social abilities. And what you can see here is that this is 6 months, this is 3, that over developmental time, the infants with later autism fell, you know, increasingly behind other children in their social skills, whereas the kids who, in green here, who had an older, typically developing sibling, were sort of keeping up with developmental expectations, which would be being on this 100 line here. So there's sort of gradual emergence of these differences in social behavior that we associate with autism later. And then we looked at, you know, some questions around how is that related to genetics. So these are polygenic scores associated with autism. So they're kind of like adding up all of those different individual genes that are associated with autism to see how many of them you have, and looking at whether that's associated with these changes in social behavior. And we do see some emerging differences, such that higher polygenic scores for autism started to become associated with social difficulties in behavior, at least at age 2. So again, this sort of emergence over developmental time on the behavioral level. And what that seems to be preceded by is this gradual developmental change, or accompanied by as a gradual developmental change in more fine-grained aspects of social attention. So this is some work that's some of you might be familiar with from Army Klin and Warren Jones in Yale, and they did eye tracking measures over the first 2 years of life in infants with later autism. And they found again that very early on, infants with later autism actually looked at faces just as much as other infants did, but then over developmental time, that percentage drops. So then by age 2, we're seeing this phenomenon we associate with autistic children in general of less looking at faces. And this is a similar graph taken from behavioral coding of developmental assessment, looking at how often children were looking at faces per minute from 6 months to 3, and again showing that same developmental change. So one interesting thing about this was that what wasn't found was what I think a lot of people expected, that infants with later autism would always have this sort of core deficit, in inverted commas, in social attention, and that would mean that they, you know, didn't look at people, didn't learn about people, and thus were behind socially on a longer term basis. Rather, what we're seeing is this initial early social skills that are equivalent to typically developing children, and then something changes over those first 3 years. And so, you know, one of the questions around that sort of trajectory then is, well, are there changes in the brain that happen that precede this developmental change in social attention? And so this is some NEARS work that was done at CBCD, trying to ask, you know, if we look at 5 months, so before infants show differences in social behavior, do we see any differences in social brain function that might precede that? And so this was work using NEARS to look at differences in the temporal lobe in response to social pictures and social sounds versus non-social pictures and non-social sounds. And there were differences in infants who had later autism who didn't show this sort of normal specialized response to social stimuli over the temporal lobe that other children showed. So maybe some evidence that that we're seeing differences in social brain function that do precede these differences in social behavior. We've also looked at similar things in the temporal domain. So Anna, who some of you will know, GUI, who looked at some EEG data, again, at sort of 5 to 6 months before we see behavioral changes, looking at neural responses to faces, and again, finding that we see something consistent to what we reported before too, that there are differences in responses to faces and non-faces in attentive responses at 6 months, that we think, again, relate to later autism. So converging evidence that around 5 to 6 months, there are differences, at least in neural responses to faces. So there are these differences in social brain function, probably social behavior, that emerge over time. But we know that this is only one tiny piece of what's happening. And those sorts of questions and I guess ways of looking at the data do come very much from this idea of identifying deficits, or at least differences that might have a sort of simple relationship to later core traits or symptoms of autism. And so what we've been doing more recently is to try to expand out beyond that, to think, well, what actually do we think is important to children in this age range? What is important from what caregivers tell us and what autistic people tell us, and where can we start thinking about this more from that sort of neurodiversity affirmative or adaptive kind of framework? And so what we've been trying to understand more recently is how some of these broader differences that we see might be related to more basic differences that might emerge earlier on, and particularly in the domain of sensory functioning. So we know that sensory differences are really common in autism. They can be really challenging for children, for providers, for adults as they grow up. And we also know that there are at least theoretically links to anxiety and to mental health difficulties, again, from people with lived experience, but also emerging empirical data. But we also know that sensory differences may have impacts on some of these other domains, like the social attention and social functioning I talked to you about before. So more recently, we've started to become interested in mapping how that might, play into this sort of more specific, neater, I guess, story about social functioning. And so when we look at sensory processing in infants with later autism, we do see these changes in sensory functioning very early on. This is some work that Elena led, again at CBCD, in some of our infants at 10 months, where we have buzzers on their feet and we play 2 buzzers close together and then a gap. And then we look at how much the infant can reduce the response to the second buzz, which is what should happen, which means that, you're kind of gating the sensory input to make sure you're not continuing to respond to something repeated. And what we see is that in the infants with later autism, we get an equivalent response to the first and the second buzz, whereas in the typically developing infants, we see this gating, so sort of lesser blueness in the second stimulus, which suggests that infants with later autism are continuing to react more than typically developing infants. And we see similar things in other modalities. So this is infants, again, at 10 months in a eye tracking task where you're looking at pupillary response to changes in light levels. And what we see is stronger constrictions in infants with later autism. So if the screen goes from black to white, their pupils constrict more. And so they need to reduce the amount of light coming in more than other infants do. And again, this is infants listening to sounds that Anna Kolesnik looked at. Again, looking at the difference between the EEG response to the first or a third sound in a train of repeated sounds. Again, you should see habituation, which would be what typically developing infants showed, whereas the infants with later autism increased their response to the sounds as they were repeated. So across all 3 modalities, broadly, it looks like greater cortical reactivity to sensory stimuli in early infancy. And so going back to that sort of, I guess, neurodiversity affirmative or sort of more adaptive way of thinking about it, the question then becomes, what differences in behavior do we see that might emerge from these differences, and where are those adaptive and where are those might be maladaptive or unhelpful for the child, and what can we do about that? And this is the journey that we're on at the moment. So what I'll describe now is more emerging data and emerging thoughts that we're still trying to work out, the potential implications of. So one thing we've been trying to do is look at how some of these things change over time. And for this work, we've used a lot of parent questionnaires, behavioral measures, to try to understand how infant behaviors that are relevant to sensory sensitivities and relevant to sort of emerging, maybe sort of mental health type differences, are interacting over developmental time. Because we know from kids and adults, there's some evidence that sensory sensitivities relate to mental health difficulties, particularly anxiety. So we're interested in whether or not even at these infant stages, you would see relationships between sensory differences and what might be a sort of precursor to anxiety, but sort of infant fearfulness responses. So this was some work led by Nisha. And this takes parent report measures of hypersensitivity, so asking questions about how much your baby reacts to sights or sounds in the environment, and then infant fearfulness, so how much do they not like to go to a new place or not like it when a new person comes. And this is using cross-lag panel models to look at the interrelation between those two, between 10 months and then 3 years, where we can measure autistic traits. And so these figures always end up being a spider's web, but the main paths that I think are interesting here are in green. And so what she found was that at 14 months, measures of hypersensitivity predicted measures of fearfulness at 2. So some suggestion that being more sensitive to sensory stimuli predicted greater fearfulness or reactivity in toddlerhood. And then also interestingly, these measures of sensory sensitivity at 2 did predict autistic traits at 3. So again, some evidence consistent with the idea that there's greater levels of sensory sensitivity in infants on a developing path to autism. But then we were also interested in the sort of other side of it. So are there protective factors or things that can help a child buffer some of these associations over time? So Mutlahan looked at again, these early measures of sort of fearfulness, shyness, that may be precursors to anxiety type behaviors. And we see this here. So these measures of fearfulness and shyness that associate with anxiety like traits at 3. But then also whether or not effortful control would be something that might buffer these sort of early associations. So effortful control measures things like how well a child can wait for something you ask them to wait for, or regulate their emotions or calm down if they get upset. And what she found was that, as I said, fearfulness and shyness predicted later anxiety and also autistic traits, but measures of effortful control also predicted later anxiety and autistic traits. If you have better effortful control, you are less likely to have anxiety or autistic traits at 3. But also interestingly, that higher levels of effortful control at 14 months-ish predicted less shyness at 2. So some suggestion that there's some combination of your early sort of tendency towards being fearful and your ability to regulate your emotions and behaviors can in combination shape this sort of emerging shyness, anxiety kind of pathway. And that might give us some ways to think about early intervention or early support through this sort of effortful control mechanism. But then to broaden it out further, we were also interested in how all of this intersects with sleep, because we know that sleep is something that in older children and adults links to a lot of these different things, and mental health outcomes, to sensory sensitivities. We all know if you have a bad night's sleep, you can be more sensitive to things in the environment. And there's some evidence that might also be true in infancy. And we also know that autistic children, one of the biggest things that parents support is that they struggle with sleeping. So we were interested in whether some of these emerging differences might either be all sort of cascading from sleep problems or interrelate to sleep problems in some ways. And again, sleep is a putative target for intervention in infancy. There are things we can do to help children with their sleep. So we were also interested in whether that might again provide another way into this sort of system. So JJ, some of you will know, looked at sleep in again our cohort of infants. And these graphs, probably this is the most important one. So this was taking measures of night sleep and doing a factor analysis to group them together. And what she finds, if you compare the infants with and without later autism, is that by 14 months, the infants with later autism were sleeping significantly less than the infants without later autism. I think it equates to about an hour and a half difference every night in terms of how much they were sleeping. So it was a really significant amount for the parents and the family. And you can see this was sort of emerging over that first year, but was really most clear at the age of 14 months. So we were interested in how this difference in sleep interrelates with some of these other things. And so one thing we did was try to understand how sleep differences might relate to just outcomes in general, so autism outcome, but also children's adaptive social behavior, so their social skills at 3 and their cognitive abilities. And so again, this is a structural equation model looking at night sleep at the top, and then visual attention at the bottom that I won't talk that much about because it's not relevant to this. But what we found again was that night sleep by 14 months associated not just with later autism, but with autism traits, with social behavior during the day, and with cognitive skills too. So we think sleep in that first year is probably really important, at least as a predictor or an associate of later developmental outcome. Of course, we don't know anything about causality yet, but we can see that there are at least these big differences emerging that do relate to later phenotypes. So then we were interested to look back to how this then relates to these sensory sensitivities measures. So, I said in the beginning, one of the reasons to look at sleep was that we think that there's the possibility that sleep is going to be intersecting with sensory sensitivities, both at the brain level, but also potentially behaviorally. And there may be some association between all these different things in early development. And so this was some work that Anna did, looking at, again, this measure of tactile sensitivity that I talked about before, where we have the buzzers on the child's foot, and we look at the gating response to the second buzz out of each pair. And so what we found in the infants with later autism was a reduced drop in response to the second buzz. And what Anna found here was that infants who were poorer sleepers, so this is a measure of sleep onset latency, which is a little bit different to total sleep, but is how long it takes you to fall asleep. But infants who took the longest to fall asleep were the ones who also showed the poorest suppression between those two buzzers at 10 months. So some concurrent relationship between your tactile sensitivity and your ability to sleep, which makes sense logically in that you imagine if you're a child that's very sensitive to the tactile sensations around you, it is going to be harder to go to sleep. But it's something that we can see in their brain measures too. So then we were interested in these sort of broader behavioral measures of sensory sensitivity. So the ones that we've been looking at from the parent report questionnaires, because we know that there are differences probably between sensory processing in the brain and the behavioral sensory reactivity, where children are explicitly showing you that they're withdrawing from different sensory sensations in the world, or they're reacting more to them than the children would. And so this is some work that, again, JJ did to model our sleep scores. This is the sleep scores at the top. Sorry about all the acronyms, not very helpful. And then at the bottom, this is a measure of sensory sensitivities, similar to the measure that I talked about before. But what JJ is looking at here is the interrelation between these sleep onset problems. So this is the similar thing to what Anna just looked at in the slide before, and these behavioral measures of sensory reactivity. And what we found when we look at that is that these sleep onset problems by 14 months predict sensory reactivity measures, so behavioral measures of how much a child is responding to sensory things in the environment at age 2. So that the harder it was to fall asleep, the more reactive you are to sensory stimuli in the environment as a toddler. And again, we see this measure with autistic traits, even though this is a slightly different sleep measure to what we looked at before. So there's some evidence that these early sensory differences in the brain are both concurrently related to sleep, and then these sensory reactivity differences in behavior are predicted by earlier sleep differences in infants with a family history of autism. So this is my extremely unclear summary picture of that story, that we're seeing these sensory differences in the brain from very early on. They're interrelating with sleep, but sleep's also predicting behavioral changes in sensory sensitivities. There's interrelations with these early anxiety, fearful kind of behaviors, and we can start to imagine that it's possible that some of those withdrawal type behaviors are adaptive reactions to the fact that if you find sensory stimuli overwhelming, then one sensible thing to do is to withdraw from that. What we need to think about is, where is that helpful for the child and where is it unhelpful, and can we change the environment that they're in to reduce their need for that. And then these sort of effortful control regulatory skills might help the child in perhaps buffering how much these sensory differences are impacting on their everyday behaviors. But one thing we're sort of interested in understanding now is, can we look at how some of these differences are manifesting on a more short-term timescale in the brain? So what I've been talking about mainly now is looking at longer-term developmental interactions, so across months of child development. But one thing we also want to look at is these sort of second-to-second effects within the brain, and particularly how do these different skills and abilities, or these different experiences, influence what we might call brain states. So epochs of time that the brain is attentive or inattentive, because that's probably on the shorter term what controls, how these experiences are influencing the development of neural connectivity, or what the child's learning, what they're not learning. So what we're trying to do now is look at some of the brain activity in a different way. So all of the studies I talked about before, we're using this measure, this event-related potential measure, where we present pictures over and over and then average together the brain response to each picture, and look at how fast on average a baby's brain responds to a face or an object. But these state-based approaches are more trying to understand what kinds of oscillatory rhythms, so what kind of brain rhythm are prominent in a child's brain activity at a certain time, and can that tell us something about the state they're in? So are they attentive, are they tired, are they inattentive, are they learning, etc. And so this is some work that Rianne's been doing, so looking at different oscillatory frequency bands and how common they are in EEG signals. And one thing that we're really interested in is theta, because that often is implicated in attention and learning. It's often greater during social interaction in children. And one thing we've looked at in the past, this is Ellie, for some of you know, is how changes in some of these oscillatory brain states might relate to later development. So Ellie looked at children watching movies and showed that changes in theta power during that movie, so children whose theta power increased, which might mark a sort of attentive learning state, were children who had better cognitive function at age 7. So there's some association between really early infant neural responses to some of these stimuli and later cognitive development later on. So we're interested now in understanding, well, can we use that to understand in infancy what different behaviors or experiences might impact those rhythms, because we might be able to then say something about how they might relate to longer term outcome. And so one thing that Louisa, one of our postdocs, has been looking at is these different changes in brain state and how they relate to infant sleep. So one thing we're interested in is it that sleep in a baby is relating to these daytime brain rhythms in a way that we might help us link it to how it's having these impacts on, or at least associations with, later cognitive development. And so this is a study that Louisa ran actually in Germany. So this is a study with typically developing infants, and this is something now that we want to bring into our siblings. But she measured infants over 4 different visits, 2 months apart, with EEG, but also actigraphy, diaries, lots of measures of sleep. And what she found was that indeed the infants who sleep worse were infants who showed less of this change in theta power when we measured them during the day. So this metric that we'd associated before with cognitive development was also associated with how well baby was sleeping at night on a sort of day-to-day, month-to-month basis. So we think this is maybe preliminary evidence that possibly some of these brain signals can help us understand, in the moment, what differences in a child's behaviors and experiences might be relevant for their longer term development. And what Louise is doing now is trying to measure some of these brain states during sleep too, so we can understand whether or not eventually infants with later autism, whether they're experiencing different brain states during sleep itself. So are they just sleeping less time, or are they also experiencing different brain states during that sleep period? And what does that mean in terms of their ongoing brain development? So just to summarize, I guess, where we've got to and where we're going, we see these really complicated interactions between these different systems. we're trying to view this through a framework of understanding which of these things are adaptive for a child and which aren't. Where might we be able to provide environmental changes that might support their development? And might that be around sleep or changing the sensory environment, or perhaps supporting early regulatory control skills? And how can we use measures of brain function to help us understand some of these relationships and help us test some of these sort of causal pathways? And the other thing we've been trying to do to support this is to do some sort of theoretical work to help us ask those questions. So this was a paper we wrote with Mark Johnson, who a lot of you will know, to try to see things from this sort of adaptive perspective and ask how can we use the kinds of data that we generate to ask questions about a child's developmental trajectories and where that may be adaptive for them and what things aren't. So I guess my overall summary is that we know that there are these really complex series of cascades that are contributing to the phenotypes that we see in childhood. And to measure that and understand that, we need to start looking at both these sort of short-term dynamics and these long-term dynamics. But the sort of underpinning thing that we need to bear in mind at all times is that whilst we want to support strong outcomes for children and their sort of optimal trajectories, you mustn't lose sight of how neurodiversity itself enriches our society. I think that's it. And thank you to our funders again and the families. Astrid, I think you're going to field questions for me, right?
I am. Goodness me, Emily, that was fantastic. Really, really enjoyed it. Your models were just fascinating. I mean, I particularly like the effort control combined with the shyness on that anxiety pathway. That was so interesting. So there are a few questions, really interesting questions, but I will use my chair privilege because I've got kind of a theoretical question. I hope it's not a weird question. So when I was doing my master's, so I did my master's in developmental disorders, but it was about 4 years ago now, and I haven't really kept up with the research since then. At that point, there was a lot of talk about multifinality and equifinality, so when multifinality, when children with like a similar starting point end up with different outcomes, and equifinality, different starting points leading to similar outcomes. But I'm listening to your talk, and it sounds to me like your kind of cascade developmental trajectories approach is kind of going beyond that framing. I mean, thinking back to, the video of the ball at the start, I mean, the ball could drop anywhere and it could end up anywhere, theoretically, shaped by the topography as it goes along. So I guess my question is that kind of multi-equifinality theoretical framing still relevant to your work, or do you feel like you've sort of moved beyond that?
That's a really good question. I guess it kind of embeds both of those elements, right? Of there being a sort of many to many kind of mapping.
Many to many, yeah.
Yeah, but that theoretic, I mean, I guess one of the ideas behind the sort of developmental cascades framework that a number of people use is that there will be these sort of canalization pathways. So although you're starting off with lots and lots of different, causal factors, there'll be certain ways in which that becomes self-reinforcing. So if you say, I mean, so the older idea in autism was that if you don't look at faces, then you don't learn about faces, and then you don't interact, etc. And that's one way in which you might get many reasons you don't look at faces, but if that converges on not looking at faces, it contributes to different outcomes. And I think probably that is one way of thinking about some of these other effects, that, if you get something that impacts your, say, confidence in interacting with new things, then you're going to be exposed to fewer new experiences. And that might, lead you into this path of sort of fearfulness, anxiety. But then you're also going to get lots of variability in how that manifests itself, depending on all of the other things that you don't struggle with. So, I think, but it's a really good question. I think there's lots of different ways of thinking about it in the field, and I don't know that we've necessarily come to a sort of coherent agreement as to what the best one is.
Brilliant. Thank you so much. I'm glad it wasn't a weird question.
No, not at all. It's a really good question.
Brilliant. Oh, okay. Well, moving to the chat, I can see we've got quite a few questions here. So let's start with Diane at the top then. So Emily, can you say more about the overlap between autism and ADHD, please? So how common is it to have both?
Yeah, so there was a long period where theoretically you weren't supposed to have a sort of dual diagnosis, although in practice that wasn't always followed. Now that's changed. So I think it's generally common to have ADHD traits, at least, or an ADHD diagnosis in autism. I think less so the other way around. So I think people with ADHD, it's less common to then have a secondary autism diagnosis. That makes sense. But we do see, fairly moderate, I guess, associations between autistic and ADHD traits in our cohorts as well. There's lots of arguments about whether or not some of the differences that might look like ADHD traits in autistic people are actually autism, not ADHD. To what extent is the etiology of ADHD in autism the same as the etiology of ADHD outside autism? So there's still a lot of discussion about it. And one thing people have started to do is look at these polygenic scores for autism and ADHD and look at the overlap within them of genetic variant. You also get a lot of single gene conditions that raise the likelihood of both autism and ADHD, so some of the genetic syndromes. So there's definitely overlap between them, but there's a lot of argument, I think, about why and what the mechanisms are.
Yeah. On that note, you mentioned polygenic scores. That's another thing I was thinking about. I mean, obviously, I guess you haven't incorporated them in your current work. Is that something you're possibly looking to do in the future?
Yeah, definitely. So we have genetic scores for most of our cohort, although there's still efforts to finish it off. So we've started to look at it. So Anna Gilly published a really nice paper looking at polygenic scores and some of the neural responses to faces. So we see differences in neural responses to faces that associate with polygenic scores for autism. And we've been looking at some more trajectories of social functioning, communication functioning. And so we do see, so like the ADHD polygenic score associates with slower communication development, whereas the autistic polygenic score associates with slower social development in our infant cohort. But yeah, there's still lots more to do. So we haven't looked so much at any of the sensory things yet, or any of the mental health outcomes. So yeah, it's a really interesting area.
Interesting. Thanks, Emily. So on to Helen. So Helen is asking about how you're recruiting the babies and young children in your research, as a lot of people are identified later in life.
Yeah, so the families who work with us are all families who have an older sibling or a parent with autism or ADHD, and then we work with their youngest infant sibling. So that's for exactly that reason, that in terms of looking at early development, we have to kind of look before children have a diagnosis, which means finding them. It is challenging on the ADHD side because, of course, a lot of children with ADHD aren't identified till they're 7, 8, 9, at which point mom's already had baby 2 or 3. So we do where families have concerns about ADHD and their older kid, if they're sort of 3, 4, 5, we do screening. But increasingly we work with autistic parents and parents with ADHD who are having who have new babies, and that's been a really fascinating cohort to work with. And I think I would say that's become much more common within our cohort over the last sort of 3 or 4 years because of, I think, partly the rise in recognition of autism in women. So yeah, that's been really nice because we get a different perspective sometimes from those families than other families.
Right. Brilliant answer. So next we've got Tracy Tokuhama Espinoza. Lovely to see you, Tracy. If anybody would like to unmute, please feel free to do so and ask yourself. But I'll just ask what you've written in the chat. So, can you talk about how you teased out poor sleep and increased sensitivity? So, for example, how do you know if poor sleep caused higher sensitivity and not that higher sensitivity caused poor sleep?
Yeah, it's a really good question. So with the concurrent measurements, we can't basically. So yeah, where we're looking at correlations within the same age group between sleep and neural responses to tactile stimuli, we don't know. So it could be that one causes the other, or the other way around, or that they're just correlated through a third factor. With the developmental models over time, we get a bit more information because we can look at what precedes what. So whether changes in sensitivity in a child precede changes in sleep, or the other way around. And what we're seeing, at least initially, is that changes in sleep precede the changes in sensitivity behaviorally. So that is at least more consistent with poor sleep causing sensitivity later on, but we still can't say that necessarily. It still could be a correlation through a third factor. So one thing we really want to do is to do some sleep interventions for these families, partly because it's a huge problem for them, regardless that their children are sleeping so badly at night. But also that is one way of getting at this kind of causal question. So if we change sleep, do they then show a reduction in sensitivity?
Great. Thank you, Emily. Okay, well, Alexandra says she has a similar question. Did you find any correlation between earlier heightened sensitivity and subsequent sleep problems? Did these studies show whether sensory issues also predicted sleep problems?
Yeah, that's a really good question. So one thing we're doing right now is coding our sleep data from 2 and 3. So what I didn't have in those models is later sleep, so sleep in toddlerhood. So that's what we're looking at now to see whether or not some of these earlier sensory differences are predicting the sleep problems later on. So yeah, we don't know yet, but we got an email earlier just saying that most of that data is now finished. So I'll let go.
Yeah. There we are. The last part of her question was, in the same way that less sleep could impact reactivity, could increased reactivity impact sleep? I mean, it sounds plausible, doesn't it?
Yeah, completely. And I think in these sorts of cascade models, that often is one of the ideas that, you sort of you have, maybe a disruption in sleep increases your sensitivity, and then you're more sensitive, so that increases your sleep. And you get into this loop where they amplify each other in this sort of positive feedback mechanism over time. So yeah, that's definitely something that we want to look at. And one thing we'd really like to do is have more nuanced data on a day-to-day basis, so that we can measure sleep with the actigraph at night, and then sensitivity during the day with some of these arousal measures that we can do, heart rate, you can do at home. So on a day-to-day basis, we could see, if you sleep more poorly, is your arousal higher? And then do you sleep more poorly? And how does it amplify on that more fine-grained scale?
Incredible. Yeah. It's fantastic. So Elena says, thank you so much, Emily. That was amazing. Liz says, is there a danger of a false dichotomy in the maladaptive adaptive distinction, particularly given how differently parents and autistic children, including when they grow up, may define something that is adaptive?
Yeah, definitely. I think that's a really important point. So one thing we've been doing recently with a group of autistic adults who work with us on a different study that's about adults is looking at some of these measures, like the Vineland, that's described as an adaptive scale. So it's about adaptive behavior, so sort of everyday behaviors, but that nature of adaptivity is defined in some parts through quite a sort of normative lens. And so Shifra, who's a postdoc who was working with us, was doing some work with autistic people to ask what they thought of the behaviors that the Vineland was asking about. Would they view that as adaptive for them? So yeah, there's definitely sort of adaptive and adaptive, right? And there's different ways to think about that. So does it mean adaptive for the kind of group in general, or for the individual themselves, or for that person's brain? And how do we define that? And actually we've been having some, I think Vinny's here, but we've been having some quite interesting discussions about how does one define what adaptive is in a way that you can then measure whether or not a particular behavior is or isn't adaptive for that person. Yeah, completely. I think it's a really important point that we need to completely figure out how to do it. And of course it's hard with young children because you can't ask them very much. So like with adults, you can ask them, for them, what would they view as helpful or not helpful, but we can't with babies. And so we have to work with parents, and as children grow up, they often have different opinions of their parents on many things, and mine do certainly. And so yeah, it is something that's really important for us to try to tackle, but it's not easy, I would say.
Thank you. It doesn't sound easy at all.
No, if anyone's got any ideas, that's mega helpful.
Thank you, Emily. Yeah, no, Liz, if you want to follow up on that, please feel free. You're happy? Okay, cool. So Ifeoma asks, is there any study you're doing with older children with autism and ADHD?
Yeah, we have a couple. So we've got a new one starting with well, they're not much older, preschoolers, autistic preschoolers, like 3 to 5-year-olds, looking at all different things around sensory processing, tactile processing. And then we have a study that we're collaborating on that's run at King's, but we do a lot of work with the EEG data, which is a study on autism, kids with autism, ADHD, and epilepsy. So children from those three different groups, but then also children who have both. So that's following children from 3 to 6. And then we there's another study that we're part of that follows kids from 6 to adulthood, not longitudinally, but sort of cross-sectional stroke longitudinal in autistic children. And one of the nice things is with some of the tasks that we do right the way through those cohorts. So some of the more naturalistic EEG kind of measures, videos, things like that, we can do across all of the cohorts. So there are questions that we can ask about development throughout that. And so in the study with older kids, for example, we're looking at sleep and sensory processing and mental health outcomes, which we obviously at that age we can actually look at sort of clinically defined mental health outcomes. So we're trying to, put some of this stuff together to understand it from a more lifespan sort of perspective.
Amazing. No, I remember reading a bit about the epilepsy and the sleep link and how it's all interconnected. Oh my goodness, that's fascinating. Thank you so much, Emily. We've got a couple more questions. I was just about to say any more questions, and we've got a couple more. So Dr. Wolf says, what are the implications for early childhood measures measuring adaptability for autistic children? Do you see having to create new norms since most measures are for neurotypical individuals?
Yeah, it's a really good question. I think there are efforts to develop new measures specifically for autistic children. So that is quite new and sort of emerging, and there aren't that many validated yet, but there's definitely efforts to do that. And as you know, I was talking earlier about, there's also efforts for intervention studies to develop sort of more person-centered outcomes where you're actually working with a child or a child and their family to, on an individual level, define what it is that they might want to get out of an intervention. So you can use vignettes and things like that. There's also this approach called normative modeling, but it's a really terrible word for it. But a sort of statistical approach to taking, say, a group of autistic people as your reference range and then defining another autistic person relative to autistic people as a group rather than sort of neurotypical or population samples.
Yeah,
it's becoming more common in studies of brain imaging. Well, we've got big enough data sets to start to do it, but it might be something that would be nice to start thinking about too on the behavioral side. So at least, you're sort of comparing children to their own neurotype rather than completely outside of that.
fantastic. Can I just double check? Are you okay to stay a couple of minutes for a couple more questions? Yeah, you're okay. You don't have to finish bang on. Zara, I can see you've got your hand up, and I think you got in a second before Cheyenne, so you can ask a question.
Hi, can you hear me okay?
Yeah.
Fantastic. Hi, Emily. Thanks for the great presentation. This question's kind of semi-formed in my head, but it's basically about these children, obviously you've been following since a very young age, and you then check their autistic behavior at quite young as well. And I'm kind of just wondering about how things like age of diagnosis or age at which these children start to present autistic traits that can be measured through the measures that we have right now behaviorally, and how that might also relate to like longitudinally how stable these predictors are. I don't know if there's anything on that in terms of like what, like how these young children will be when they're like 5, 6, 7 or older.
Yeah, it's a great question. So yeah, we are doing that right now. So we've just finished doing a follow-up of some of the kids who are in our older phases, who are now like 6 to 12 ish. So we've just finished that and looked at all of the data, and yeah, we definitely see that almost all of the children that had a diagnosis at 3 also had a diagnosis at 6 to 12, but there's also another basically 50, another same number of kids again who by 6 to 12 meet criteria for autism and just didn't at 3. Some of them had some early traits, some of them didn't at all. So that's really interesting. So yeah, one thing we've just pre-registered or about pre-register is looking at then these early infant markers and how they relate. So we know that some of them relate to diagnosis at 3, but then what did they look like in this group of children that didn't meet criteria till 7? Do they have like this later emerging kind of phenotype in sort of neurally as well as behaviorally, or Or do we see something completely different in those children? So yeah, it's a really, really interesting question. But yeah, one interesting thing is that certainly the kids who, and it kind of makes sense clinically too, that the children who by 3 that you're confident enough to say, you know, they're autistic, they still are later on. But yeah, there's this whole other group that really you couldn't tell earlier.
Brilliant. Thank you. Zara, are you happy? Yeah, good. So I think this will be our last question from Shen, and it's a really interesting question, actually. So have you compared male and female outcomes and data, you know, obviously with females being better at masking, you know, etc.
Yeah, no, it's a really good. We haven't done enough actually on that in our cohorts on sex differences. There's definitely, often we're just not quite powered enough, and it's taken us a long time to get to the point where we've got the power to start to ask about that. And we've looked at, so Teresa, who's a postdoc at CBCD, has done a lot of work in our older cohorts looking at sex differences in things like social attention and masking kinds of behaviors. And we do see lots of interesting patterns there, but we just haven't done enough yet in the infants. There are some trends so I think we see more girls, for example, that are in this sort of later diagnosed group that we recognize later on, which sort of fits with what you see clinically. But yeah, it's definitely a very good question, and we do have lots of data and not enough people to analyze it. So if anybody wants to come and join us with a research project on sex differences, that would be awesome, because it's something that I really want someone to focus on, and we just haven't quite had the time. So yeah, do let me know.
There we are.
Wants to come?
Brilliant.
Yeah.
Casting call. Brilliant. Oh no, thank you so much. As Julie says, lots of food for thought. I feel like my brain is like popcorn right now. It's just going off everywhere. So thank you so much, Emily. Really, really appreciate your time and your expertise. Absolutely fantastic presentation. Yeah, I guess that's it for this evening. Thank you so much, everybody, and we'll see you at the next one.
Thank you guys.
Bye, everybody. Take care. Bye bye.